Hey Lauren, what's going on?
I'm trying to review what happens in the cell cycle, but I'm not sure if I remember all the cool stuff that's happening.
Well, let's review it together. I can't get enough of the cell cycle.
When your time comes, don't postpone it. When others doubt and out, you don't condone it. Truth be told, yourself is your toughest opponent. When your moment comes, grab hold and own it. Never let go, stand tall and hold tight.
But before we talk about the cell cycle and its series of stages and how it gives the genetic instructions for characteristics from parent to daughter cells, can you quickly remind me of the differences between gametic and somatic cells?
Of course I can. We definitely should make sure we're on the same page for the basics. Remember, our gametic cells are our reproductive cells, such as egg and sperm, and they're associated with meiosis. While our somatic cells are all of the other cells, such as skin and hair, and that's associated with mitosis.
So what do you remember about the cell cycle?
Well, I know it has two major phases, interphase and the M phase.
And the M phase stands for mitotic.
That's right. And I know the interphase is the period between cell division in which the cell grows, develops, and functions. And interphase is divided into three stages, G1, S, and G2.
And I know that the mitotic phase includes mitosis and is the process of nuclear division and cytokinesis.
You got it. But there's so much more going on in each of these major phases. We should go into more detail. Details are always the best.
That was my idea exactly. Well, let's start with interphase. I know the first stage of interphase is G1, and in G1 the cell grows and proteins needed for cell division are synthesized, and at the end of G1 there is the G1/S checkpoint.
Really? What does the checkpoint do?
Yeah, the checkpoint's really cool. It ensures that all cellular components are there and everything is in good working order, so any cells that are damaged or missing chromosomes do not move on.
Okay, but what about G0? I remember a G0.
Oh yeah, I almost forgot. G0 is just a stable state, or a non-dividing phase. Cells can stay there indefinitely or re-enter the G1 active cell cycle.
Okay, so G1 stage is really just the time for cell growth and development and check cells to make sure that they're all good to go to the next stage.
Yep, you got it. Then the cell moves on to the S phase.
I know all about the S phase. It's all about replication and DNA synthesis, right? DNA synthesis is really important. It needs to happen before the cell can proceed to mitosis. What's really cool about the S phase is that before the S phase, each chromosome is unreplicated, and after the S phase, each chromosome is composed of two sister chromatids.
Say what? I forgot that at the end of S phase, two sister chromatids are composed, and that's really important information. But I do remember what comes after the S phase, and that is the G2, where it's all about division preparation. In this phase, there's a bunch of biochemical events happening, all for cell division. And this stage also has a checkpoint, the G2M checkpoint. And to get through this checkpoint, the cell's DNA needs to be completely replicated and undamaged. After this checkpoint, the cell is ready to move on to the second phase of the cell cycle, the M phase.
Cool. Well, let's do a quick test on the stages of interphase first. And if for any reason there are other people listening to our conversation, they should play along too. What stage has replication and DNA synthesis?
Oh, I know this one, S phase. And what stage is all about cell growth and development?
That's easy. That's the G1 stage. And the G1S checkpoint makes sure that all cellular components are there and in good working order. Lindsay, do you know what stage is all about division prep?
Girl, I know that's the G2 stage, and there's also that G2M checkpoint, and that checkpoint makes sure that all the DNA is completely replicated and undamaged.
Well, I think we have the interphase part of the cell cycle down. Do you want to talk about the mitotic phase now?
Do I want to talk about the mitotic phase? That's like my favorite phase of the cell cycle. It's when the copies of the cell's chromosomes separate and the cell undergoes division. Could it get any more fun?
Yeah, it's super cool that the M phase is made up of the 5 states of mitosis and cytokinesis.
You know I want to talk about that. And what we need to talk about first is prophase. When cells enter prophase, the chromosomes are all condensed, and each chromosome has 2 chromatids.
I did know that, and my favorite thing about prophase is that the cytoskeleton breaks down and the mitotic spindle begins to form, which is just an organized array of microtubules that move chromosomes in mitosis.
I know what happens next, prometaphase. This phase is neat in that the nuclear membrane starts to disintegrate and the chromosomes form kinetochores.
Can you remind me what kinetochores are? I always forget.
For sure, kinetochores are a multi-protein complex, but really they are just the anchor point for microtubules. And in this phase, the microtubules enter the nuclear region. They were outside the nucleus before, and the microtubules grow and shrink until they attach to the kinetochore.
Right, and after that, the chromosome becomes anchored to a microtubule from opposite spindle poles.
Of course, and this state is completed when microtubules all have attached to kinetochores.
Then comes metaphase. I know this stage really well.
Preach, girl!
In metaphase, the chromosomes become arranged on a single plane, the metaphase plate. This is between the 2 centrosomes, which are the tiny organelles responsible for arranging the microtubule network.
That's right, those chromosomes align in the middle, and at a spindle assembly checkpoint, make sure the chromosome is aligned on that plate and attached to the spindle microtubules, but from the opposite poles.
And then comes that crazy anaphase.
Really? What makes it so crazy?
Maybe that the connection between sister chromatids breaks down and the sister chromatids separate. That marks the starting point of anaphase.
And then, after that, the chromosomes start moving toward the opposite spindle poles. That is pretty crazy.
Then there's the final state of mitosis.
I know, telophase. This state begins with the arrival of the chromosomes at the spindle poles. And I think this next part is even more crazy than anaphase. The nuclear membrane reforms around each set of chromosomes.
You know what? I think that's even more than crazy. That's sick. That reforming of nuclear membrane means the production of 2 separate nuclei within the cell. And simultaneous to telophase, cytokinesis is occurring. That cytoplasm is dividing.
There is just so much going on in the M phase. I think it's time for a test.
Bet, go for it.
When do the chromosomes align in the middle?
Metaphase.
Okay, but that was really easy. When do the spindles begin to form?
Come on, prophase. Are you going to give me a challenge?
All right, you asked for it. What state has the disintegration of the nuclear membrane, and what state has the nuclear membrane reforming?
Prometaphase, that's the disintegration, and then telophase has the membrane reform.
Okay, Brainiac, what state has the chromosomes separate and move to opposite spindle poles?
Anaphase. And I think I might be a mitosis genius now.
It sure sounds like it. Now that we've went through the cell cycle, I think it would be good to just review the difference between mitosis and meiosis, because I know the next time we hang, I want to just talk about meiosis.
Sounds good. I'll take mitosis. So in mitosis, we go from 2N to 2N, and it's the division and replication of somatic cells. The nucleus of eukaryotic cells divides, and the result is 2 daughter cells that have the same number and kind of chromosomes as the parent.
So a good example of this would be skin tissue growth. While meiosis, we go from 2N to 1N, and meiosis produces gametes for sexual reproduction through meiosis 1 and 2, which is what you're going to talk my ear off about next time.
I can't wait until next time.
The only thing I want to discuss before next time, and a good way to end the cell cycle, is determining the amount of chromosomes and DNA molecules throughout the cell cycle.
That sounds like it could get confusing.
Nah, I think we can do it.
Okay, let's start with G1.
Sounds good. In G1, the cell is diploid with 2 complete sets of chromosomes.
So a total of 4 chromosomes for G1, and each chromosome consists of a single molecule of DNA.
So a total of 4 DNA molecules. In the S phase, at the beginning, the sister chromatids are still tethered together and share a single functional centromere. And at the end of S phase, the cell still contains 4 chromosomes, but each have a sister chromatid. So 4 times 2 equals 8. 8 DNA molecules are present at the end of S phase.
That sounds right to me. And for G2, prophase, prometaphase, and metaphase, the cell still has 4 chromosomes and 8 DNA molecules.
But in anaphase, the sister chromatids separate and each has its own functional centromere. So this state contains 8 chromosomes and 8 DNA molecules.
And this stays the same until after cytokinesis, where the 8 chromosomes are distributed equally between the 2 daughter cells. Each daughter cell has 4 chromosomes and 4 DNA molecules.
Just like at the beginning of the cell cycle.
Wow, that was a lot. We definitely need to save meiosis for another day.
Whew, you read my mind. See you next time to discuss meiosis.